see now.
Yeah.
Okay. Yes. So I'm going to continue with this theme of evolutionary trees that Milford talked about. Darwin, in his Origin of Species, actually only presented one tree, and it was completely hypothetical. And the reason why he did a hypothetical tree, whoops, sorry, I'm like Milford, I'm catching on here. Okay, was that he really didn't feel there was enough data. And this was also reflected many years later when he wrote his second great evolutionary tome, The Descent of Man. In this, there were no evolutionary trees at all. Although when we look at Darwin's notes, he did sketch out many evolutionary trees, including this one that showed the evolution of humans and other primates. But he refused to put any of these evolutionary trees into his publications because he felt that there wasn't really enough evidence to construct a meaningful tree at his time. They were all speculative, in his words. But others were not inhibited by the sparseness of the data. And Milford, in fact, emphasized Ernst Haeckel, who was an early champion of Darwin and evolution. And he had no problem in publishing lots of trees. In fact, by 1868, even before The Descent of Man, he published a tree showing two human races as separate tree branches. And as time went on, he kept increasing the number of races in his trees. By 1898, which was where this diagram came from, Haeckel had 12 distinct lineages of humans that he regarded as separate species. Now, Darwin strongly objected to this, and both in personal correspondence with Haeckel, but also when he wrote The Descent of Man. Darwin, particularly during his time in Brazil on his voyage of the Beagle, he knew directly that there were no barriers to interbreeding among the races. And he said in The Descent of Man, going against Haeckel, quote, but the most weighty of all the arguments against treating the races of man as distinct species is that they graduate into each other, independently in many cases, as far as we can judge of their having intercrossed. Therefore, he felt that Haeckel's trees were wrong to treat races as separate branches, and he expressed this directly to Haeckel in his correspondence. So now let's skip 100 years and fast forward to the second half of the 20th century. One advance had been made. By that time, there was beginning to be a consensus that biological races were just a synonym for subspecies. And during the evolutionary synthesis, Ernst Mayr, a very famous systematist who contributed to that synthesis, has spent a lot of time defining subspecies in a kind of a pre-genetic environment. And he said that subspecies has to have sharp geographical boundaries, sharp morphological differences associated with these boundaries. And of course, nowadays, we would add sharp genetic differences have to exist across these geographical boundaries. Mayr also insisted that subspecies has to have taxonomically diagnostic morphological characters that all members of the subspecies share and that are not shared by the remainder of the species. And this is generally interpreted in modern genetic terms as that the species is subdivided into genetically distinct evolutionary lineages. That is, they're taxonomically identifiable as separate lineages within a species. So that's the more modern concept of what we mean by a race. Now, during this second half of the 20th century, there were several models of human evolution, and here's three of them. And you've seen these before, both yesterday in Stone King's presentation and Milford's presentation just before mine. You have the out of Africa replacement model and the candelabra model. Now, these two models have no role for gene flow or admixture. Everything is descent. Everything is due to branches. And both of them, from a topological point of view, are identical. That is, they each treat the major races of humans as separate branches on a tree. The main difference between the candelabra model and the out of Africa model is that in the out of Africa, there was anatomically modern humans evolved in Africa relatively recently and then replaced all the archaic populations in Europe as they expanded out of Africa, so that the branch lengths leading to the modern races are relatively short. Under the candelabra model, they go all the way back to the early Pleistocene as separate and independent lineages. Now, in contrast to them is the multi-regional model, which we just heard about, which basically says as humans came out of Africa, there was no tree at all. There was rather a trellis because of interchange between all these populations. And there was some degree of genetic diversity among these regions because gene flow was limited by isolation by distance and ocean barriers and so on. And there was also local adaptation. So the multi-regional model did not mean that all populations were identical, but it meant there was a single evolutionary lineage. There were no separate branches. So the out of Africa replacement model and the candelabra model were kind of the intellectual descendants of Ernst Haeckel, whereas the multi-regional model was much more consistent with what Darwin had said in The Descent of Man. Now, things really changed a lot. This just shows, I want to explain this because Milford already did a good job of explaining about this network. So we don't need to portray human evolution as a tree. We have the alternative of portraying it as a network. So I won't say anything more about this since Milford did such a great job. I will go, though, back to some talks that he gave in 1932. Weidenreich argued that humans had always migrated, that their subdivision into races was purely conventional, and that characters varied in gradients, not restricted to one group, and were often due to factors other than race, such as sex, age, and environment. And hence he concluded in 1932 that races were fiction. Ironically, in 1934, he had to flee Germany because of the racial theories of the Nazis who had just came into power. So along came the Kahn et al. Paper in 1987, and this really had a big impact on much of the work that was being done. You saw this tree yesterday in Mark Stoneking's example, and the important thing is this is a tree of mitochondrial haplotypes, not of human populations. But it showed that there was a series of haplotypes. There was a major clade or branch going back to the common ancestor that were strictly African. And then there was everything else, which was found both in Africa and the rest of the world. And they also dated this, and the date was about here. It was just shortly close to this out of Africa split shown in the out of Africa replacement model. And so that eliminated the candelabra model. That was just, you only had a common ancestor going all the way back to almost the beginning of the Pleistocene. And so they said their haplotype data from mitochondrial DNA eliminated the candelabra model as a possibility. But they actually went on beyond that, because in their paper, they said the out of Africa replacement model could be contrasted to an alternative model. And means it's a singular model. And they gave two references to this alternative model. One was Carlton Kuhn, who was the primary person who was advocating the candelabra model. And the other one was to a paper by Milford Walpoff, who we just heard, which was an updated version of Weidenreich's multi-regional model. So they equated the multi-regional model to the candelabra model. And as a result, they said they had falsified the candelabra model or the multi-regional model as well. But, and this is a big but, these two models, the candelabra model and the multi-regional model, are not identical. In fact, they are the most extreme opposite models of human evolution you can imagine. This one has, the candelabra model only has evolution, independent evolution of isolated branches. The multi-regional model says there were no branches within humanity. There was always gene flow, and so all of humanity evolved as a single evolutionary lineage. So there's no gene flow, role for gene flow or adaptation in the candelabra model. There's extensive role for it in the multi-regional model. And in fact, the multi-regional model is completely compatible with the mitochondrial DNA haplotype data, because even a recent common ancestor for mitochondrial DNA could spread throughout all of the world through gene flow. So in fact, Kahn et al. Did not eliminate the multi-regional model as was claimed and many people thought. The multi-regional model and the out-of-Africa replacement model were equally compatible with their mitochondrial haplotype data. And this situation kept existing for quite some time. There was a lot of debate about the out-of-Africa model and the multi-regional model. And the first statistically significant rejection of the out-of-Africa replacement model was in a paper that I published in Nature in 2002. And this was before the ancient DNA data came out. So this was using a technique called nested clade phylogeographic analysis, which looked at areas of the genome that had low recombination where you could construct haplotype trees. So it included mitochondrial DNA and Y-DNA that Stone King talked about yesterday, but also other regions of the human genome where you could construct haplotype trees. And the nice thing about nested clade analysis is you don't depend upon any prior model. Rather, I developed a statistical framework that would test null hypotheses. So everything that emerges from it is something that wasn't created a priori, but emerged directly from the hypothesis testing framework of this analysis. And this hypothesis testing framework was confirmed as being valid by looking at 150 examples of cases of other species where we knew what happened and it worked beautifully. Also, computer simulations, which were done actually to discredit the model, but actually when they were applied to the actual framework that I had developed, showed that, in fact, this method has very high power in determining fragmentation events, that is, lineage splitting events. And it also does not yield high false positive rates, either for fragmentation events or for inferring gene flow. So both computer simulations and worked examples have validated this model. So as you can see, starting at the bottom, first of all, after we detected an out-of-Africa expansion of Homo erectus with this analysis, and then we tested the null hypothesis of no gene flow between Africa and Eurasia in this early Pleistocene part, and we cannot reject that null hypothesis. So I'm not showing any gene flow connections at that point. But notice the P level was still 0.11. So there was some evidence of gene flow even back in the early Pleistocene, but not enough to be significant. And like I say, I'm also trained in statistics. I've got a graduate degree in statistics, and I take this hypothesis testing approach very seriously. So I only show things that are supported by statistical analysis. Now, then in the mid-Pleistocene, there was an out-of-Africa expansion, not the one that's the out-of-Africa replacement expansion, but an earlier one. And here, this also occurred. This was significantly different from the Homo erectus expansion. And I can also notice that in this diagram, the lines, the lineages in Europe and Asia are not broken by this expansion. And that's because I tested the null hypothesis of no admixture. And for this early mid-Pleistocene expansion of humans, you can reject that null hypothesis at a P level less than 0.035. So it's significant, but just barely. But even then it says, when humans moved, they interbred. They didn't replace one another. But more importantly, after that mid-Pleistocene expansion, I can now reject the null hypothesis of no gene flow with a P level of 0.013, going all the way back to the mid-Pleistocene. And it's what you do is you reject it in favor of an isolation by distance, also with some resistance, particularly due to water bodies, but also that there was some long distance dispersal. So gene flow patterns have been established since the mid-Pleistocene, according to this analysis. Then the next thing that occurs in this is another out-of-Africa expansion event, and this is the one associated with anatomically modern humans. And here, once again, you notice it doesn't break the lines of descent because there was admixture. And here you reject the null hypothesis of no admixture at a P level of 10 to the minus 17th. The genetic data even available in 2002, not using ancient DNA, was so strong we could completely eliminate the out of Africa replacement hypothesis. There was no out of Africa replacement, and we've known that conclusively since at least 2002. And after that, there's more gene flow, there's a few more population expansion events, but always coupled with admixture and interbreeding. And the one thing that is absent in this whole diagram is there was no significant fragmentation events that would lead to separate lineages in humans since the mid-Pleistocene. Maybe there were some in the early Pleistocene, but certainly since the mid-Pleistocene, there were no separate lineages of humanity anywhere in the world. But there was significant gene flow, both due to isolation by distance, isolation by resistance, and long-distance dispersal. So in other words, there were no races because races demand that we have separate evolutionary lineages within the species, and there are none. So the ancient DNA data, of course, actually confirmed strongly the model that I published in 2002. And we've talked about that both yesterday. I'm just going to talk about one paper, the most recent one that used the ancient DNA data, and that's the Ragsdale et al. Paper that came out recently in Nature. And here's a diagram you can see on the bottom here, the one diagram they have. And they showed a mid-Pleistocene out of Africa expansion event. And they showed as indicating that the Neanderthals were descended from that. So they confirmed that mid-Pleistocene expansion event that was first detected by nested clade analysis. And also you see the dotted line showing admixture occurred, not replacement. But also you see all these little horizontal lines indicating gene flow. However, when you actually read the text and look at the tables and the additional material that gives all the statistical nitty-gritty, you actually find that their analysis of this combined modern and ancient DNA data is actually more strongly multi-regional than even this diagram suggests. So here's just some quotes from their paper. Allowing for continuous migration between the stem population substantially improves the fits relative to zero migration between stems. With continuous migration between stems, population structure extends back to more than 1 million years ago. That's completely consistent with the nested clade analysis. Migration between the stems in these models is similar to the inferred migration rates between connected contemporary populations over the past 50,000 years. In other words, the amount of gene flow was not small, it wasn't sporadic. There was extensive gene flow going all the way back to the mid-Pleistocene. That's what the ancient DNA data says. That's what nested clade analysis said 21 years ago. Now they further go on to say the best fit models presented here include reticulation and migration between early human populations rather than archaic hominin admixture from long isolated branches. In fact, they pointed out there are no long isolated branches. The branches do not exist in human evolution since at least the mid-Pleistocene, according to their analysis. And so once again, there are no races. They then go on to say elements of both recent expansion and African multi-regionalism feature in our best fit models. That is, when they just look at the African populations, they found multi-regionalism there in the very sense that Weidenreich used. And so they concluded that, in fact, gene flow and admixture within Africa was critical for the evolution of anatomically modern humans. And then when these anatomically modern humans expanded out of Africa, there was additional gene flow and admixture, which continued to accelerate human evolution. So admixture, gene flow, multi-regionalism is the dominant mode since at least the mid-Pleistocene, both from nested clade analysis and from ancient DNA studies. Now, interestingly, Ragsdale did draw these trees. So you have a tree with big, thick stems, and then you have the gene flow and admixture events as these little tiny lines. And drawing trees like this and adding gene flow and a few interconnecting lines is very commonplace in the human evolution literature. And in fact, many approaches to statistical phylogeography start with the tree and do this, then add admixture and gene flow as needed. And this approach has been shown to be statistically inconsistent. That is, tree first, gene flow second. And what I mean by statistical inconsistency, once again, I'm a statistician. A statistically inconsistent method means that as you get more data, with probability 1, you will get the wrong answer. It's one of the worst properties any statistical test can have, is to be inconsistent. And so what this means is if there was any gene flow or admixture at all in human evolution, this tree first approach and adding gene flow second will, with probability 1, give you the wrong answer. Okay, this is terrible, and yet this is the dominant mode that people use to study human evolution today. And in particular, we know that that method will consistently underestimate the role of gene flow and admixture in human evolution, because you start with the tree. And there's no need to do it now. We have network approaches that allow you to do a statistically consistent method of inference, and you don't need to depend on the tree. A tree, by the way, is an example of a network, but a very sparse network. So if a tree is true, you will find it with a general network approach in a statistically consistent fashion. But unfortunately, in human evolution, people depend again and again on statistically inconsistent tests, and it's really messed up the field tremendously. So you must test for treeness. If you want to draw a tree, you have to ask the question, does the underlying genetic data fit the constraints of a tree? And fortunately, there are tests for doing that. So for example, here is a tree that was published in 2016 by Hunley et al. And they estimated this tree of modern human populations by neighbor joining. But then they went back all the way to 1975, where Cavelli, Sforza, and Piazza suggested a test statistic for treeness. And so this test has been around for a long time, but nobody used it because it was so computationally complex. Even Cavelli, Sforza, and Piazza couldn't use it except for maybe a tree of 5 taxa, nothing more. So the test languished until now. Our computer power is now finally big enough that we can start testing trees with this test statistic. So what they did, and Hunley et al. Did, they used neighbor joining to get an initial approximation to the tree, and then they looked at alternatives and calculated the test statistic and found the one that minimized the test statistic. And so this is a neighbor joining tree that has been modified to minimize the test statistic of Cavelli, Sforza, and Piazza. And since it's minimized, that means it's biased in favor of the null hypothesis of being a tree. That is, treeness would be accepted. What is fascinating about this paper is that even though they calculated this test statistic, they actually never used it to test whether or not the tree that they end up with actually fits the null hypothesis of treeness. So since they didn't do it, I did it. And here are the results. Look at these P levels. In my computer, I set my lower limit as 10 to the minus 200th. I didn't think anything would ever get below that, but it did. Okay, the total tree is rejected at a level of 10 to the minus 200th or below. And all the subtrees that they looked at in their paper, none of them are even remotely fit the data. This is such an overwhelming rejection of the null hypothesis of treeness that no scientist should ever say, oh, this is a representation of human evolution. There is no tree here. There is no subtrees here. The tree hypothesis is completely eliminated by these very low levels of significance. On the other hand, you can take the same data that this was based on, and here we plot it against a genetic distance plotted against a waypoint distance. This is an isolation by distance plus resistance, where we say water barriers are more resistant to movement. And the fit of the data to this gene flow model of isolation by distance and resistance is excellent. Whereas we just saw the fit of this to a tree is horrendous beyond belief. And in fact, you notice one very peculiar thing about this tree and other trees you've seen in the human evolution. If you ask what's the total branch length from a current population to the common ancestor, you notice it's very small here, but here it's very, very large. It's just reflecting geographical distance. This tree is a complete artifact. It is not a tree at all. It is only when you force a tree out of an isolation by distance and resistance model. So you would say, oh, the literature would say, okay, we'll favor the gene flow model and not publish a tree. Like Darwin said, you shouldn't publish a tree unless you have substantial evidence for it. Here we have substantial evidence against a tree, yet these kind of trees are in the literature all the time. Not one single tree you see of human populations has any statistical validity whatsoever. So Darwin himself really won out, I think, in this match. There are no races of man because they do graduate into each other and they do intercross. So Darwin was right, and we now know, as we've thoroughly sampled the globe, much more than Darwin could in his voyage in the Beagle, we've shown this is true. I'll just give a couple of examples. There's probably no morphological trait more used to define races than skin color. But does skin color have strict geographical boundaries and sharp differences? And the answer is no. We now know, as we look across the globe, skin color varies gradually. And in fact, it varies with latitude. So dark skin tends to dominate in the tropics. Light skin tends to dominate in the high latitude areas. And we know why this occurs. Melanin protects us against UV damage, ultraviolet radiation damage, which is most intense in the tropics. And that's why we have dark skins in the tropical populations. On the other hand, you do need some UV to penetrate your skin to help synthesize vitamin D. And without vitamin D, that can lead to a whole host of very severe clinical problems. So as we get to the higher latitudes, we have light skin in order to ensure the synthesis of vitamin D. So these are not racially defined traits here. This is just local adaptation, in this case, a clinal adaptation to absolute latitude. We now know, of course, the genetic basis of skin color. The skin color genes have been mapped, and a few years ago, a group I was working with, we published a paper that appeared in Genes, Genomes, and Genetics. And we used a computer program made by a former graduate student of mine, Charlie Clymer, that can look for co-adapted complexes embedded in the human genome. And one of the complexes that emerged was a complex that included several of the skin color genes and the VDR gene, the vitamin D receptor locus. Each of these individual genes often showed a very complex pattern with latitude. But when we identified the co-adapted haplotype that put these together, that was under selection, it fit a linear model with absolute latitude beautifully. So in fact, there's no breaks here. We don't have sharp geographical disjunctions or borders. Skin color varies continuously at the phenotype level, and it varies continuously at the molecular genetic level. There are no racial traits here in skin color. Now, sometimes people talk about genetic diseases showing they're very important for race. And of course, the one most often is sickle cell anemia. And sickle cell anemia is found in very high frequency in many African populations. But you can see it's also found in high frequency in some Indian populations. Here's some in the Middle East, including Iran, and in the Mediterranean regions. The common theme behind these regions is that these are regions that either today or in recent history had high degrees of falciparum malaria. So sickle cell is an adaptation to malaria, and it reflects, its geographical distribution reflects not racial variation, but its local adaptation. So now we can conclude from all of this the following. Whoop. Okay. First of all, there are no distinct lineages within the human species. We know since the mid-Pleistocene, human evolution has been dominated by gene flow and admixture to such a degree that the concept of distinct evolutionary lineages is just completely wrong. It's falsified almost beyond belief when we apply statistics to it. And I think one of the greatest detriments in modern studies of human evolution is the fact that scientists don't use hypothesis testing. We can test a lot of these hypotheses, and if you want to publish a tree, test for treeness. I guarantee you the results will show that it's not a tree at all if it's humans. But we don't test testable hypotheses, and we need to do so, or more so. And once you do that, you realize there are no distinct lineages within the human species. There's also no sharp phenotypic or genetic boundaries between human populations. That is not to say that there aren't differences, there are. But, and I didn't have time to talk about this, but humans overall are one of the most genetically homogeneous species of terrestrial vertebrates on the planet, despite our global geographical range. So, for example, I actually just got back from the Middle East where I worked on Equus hemionus, the Asiatic wild ass. It shows much more geographical variation within in Asia than humans do in a global level. I worked on lizards in the Ozarks, a small area of the United States. They show far more genetic differentiation than humans do at a global level. So these minor genetic differences that we do find among human populations are best explained by a combination of recurrent gene flow, constrained by distance and resistance, and local adaptations. And some of these local adaptations are clinal, like we saw with skin color. Some are due to local environmental conditions, like we saw with sickle cell anemia. So there are genetic differences between different geographical regions of humans, but they're not the kinds of differences that define races. So we can conclude without any doubt, using a hypothesis testing framework, that there are no races within humans. And so I thank once again the organizers for allowing me to speak, and this has been a great symposium, and I thank you very much.